As all the audience knows, the fundamental function of ion channels is the selective ion permeation and gating. I'm interested in the elementary property of this channel function, but there is a problem. The membrane is not simply a supporting material, but an interactive environment for channel performance. But the biomembrane is so complicated. This is a recent diagram of the modified fluid mosaic model. And also, Engelmann says the membranes are more mosaic than fluid. The patch clamp-based recording this complicated membrane. We need to do the experiment with a simple membrane, and because the membrane, people do the patch clamp with a given membrane composition, and many membrane proteins exist on the membrane. We want to do the experiment with the defined or arbitrary lipid compositions and also purified channels. This is a channel reconstitution method. There are many reconstitution methods. We purify the channel and incorporate the membrane in different kind of the membrane. Also, we use FM to see the channel structure. Mainly, we use the KCSA potassium channel because it is a prototypical channel, and amino acid residue only 160, and forms a tetramer as an active form. This is pH dependent. When the intracellular pH becomes acidic, then the gate opens. This is well known, and there are many kinds of structural data. In the selectivity filter, each ion resolved, and when the ion permeate through the selective filter, they permeate as a single file. This is also a very interesting story. But to do these elementary process of ion channel, we need to use a membrane, and for the permeation property, we use simple lipid bilayer. Unfortunately, I don't have time to show this result, but you can see this paper. And in the channel lipid interaction, we need to make a complicated membrane with defined and asymmetric membrane. Through this method, we find the lipid sensor. The N-terminal MO helix senses the lipid composition of the inner leaflet of the membrane. And also, we show the cholesterol action on the channel, KCSA channel. We are not performing the experiment in the biomembrane, but this clustering dispersion dynamics is a supramolecular phenomenon of ion channel, which can be performed using this lipid bilayer membrane. Today, I'll talk about these two topics. As you know, we reconstitute the channel into the preformed bilayer. This is a classical chamber method, and we form the bilayer beforehand, and the purified channel is incorporated in the membrane. Then the channel is embedded in the membrane, which is similar to the biomembrane, except for one thing. The lipid composition is completely regulated in this reconstitution method. Across the membrane, we can apply the voltage, and you see the single channel recording. So this method has been used for a long time, but we now developed and modified this classical method. And monolayer technology is a method to manipulate the bilayer membrane in monolayer basis. So we call it contact bubble bilayer method, which is a hybrid method between classical chamber method and the patch clamp. The method has already developed by Phonax group and the Berries group in 2006 and 2007. Basically, the membrane is formed in the oil phase, which is revolutionary technique. We developed a similar technique, but combined these two methods. This is a contact bubble bilayer method. Let me explain the detailed method. We can make a soap bubble by blowing the air. You can also make a water bubble by just blowing water. But this is only possible in the space lab. But you can do the same experiment in this environment. If we blow the water in the oil, then we can get the bubble like this. This glass pipette is similar to the patch pipette, but the diameter is about 30 micron. Big pipette is used, and this diameter of the bubble is 50 micron. Once the bubble is formed, the phospholipid is accumulated at the water-oil interface, forming the monolayer. Once two monolayers are attached, the bilayer is formed in this region. This is a contact bubble bilayer method, which is very conveniently manipulate bilayer in a monolayer basis. Let me show the method conceptually. This is a usual liposome. Our method is just conceptually peeling down the outer membrane and form another bubble. So this is a liposome peeling out. This is the bubble, and making the bilayer region here, and the other part is lined by monolayer. So this is just a topological transformation from the liposome to the two droplets. Using this method, we addressed the question of the lipid sensitivity of KCSA channel. In crystal structure resolved in 2001, the lipid is co-crystallized with KCSA channel, and this lipid is solved as phosphated glycerol. PG is rich in bacterial membrane, and this PG is located at the outer half of the transmembrane domain of KCSA channel. So people suspect this PG should be very important for the channel function. To do the experiment, we formed the membrane with PG, PC, or PS or PA. We can make the pure lipid membrane and incorporate the channel. You see, the channel opens almost all the time. The open probability is about 90% when the channel is incorporated PG. But when the membrane is formed by PC, pure PC, the getting open probability is about 10%. Such big differences in the channel gating occurs by changing the lipid composition. These are the other lipid. These lipids are anionic lipid, and these are neutral. And this is another kind of phospholipid, but with positively charged. So by changing the head groups, charged condition of the head group, the single channel recording shows different types of the activity. Almost 100% open at these negatively charged lipid, but only 10% for the neutral PC, but the open probability is much less than this channel, less than 10% for positively charged EPC. So anionic phospholipids keep the channel in the open state. You see the different conductance. This is simply because of the surface potential. But we focus on the gating property of this lipid effect. And the next question is, which part of the inner wall outer leaflet should be important for such a different lipid composition? This is simply a freeze fracture, but this is irreversible steps to making the two leaflets apart. But in this case, we can repeatedly form the bilayer or monolayer with the totally asymmetric membrane composition. In this case, liposomes with defined lipid composition is incorporated in the bubble. In this case, this is simply PG, liposome with PG, and in this bubble, the liposome contains PC. And then two bubbles are connected. Then the totally asymmetric membrane is formed. And the flip-flop of the lipids is very infrequent. During the recording, we never see the flip-flop. I mean, a couple, 10 minutes or something. And when the inner leaflet, you see, this is a cytoplasmic domain, so this is inside, cytoplasmic side. If the inner leaflet is PG, then the channel open probability is about 90%. But if the inner leaflet contains PC, the open probability is much less. So the PG should exist in the inner leaflet of the membrane for the high open probability relative to the PC in the shows very small open probability. So people expecting the outer part of the transmembrane region should be important for the lipid interaction with the channel, but as far as this channel activity gating, this outer leaflet crystally resolved, this PC is not important for the activation gating of the KCSA channel. Now the PG is a negatively charged at the head group of the lipid. It should interact with the channel molecule electrostatically, and there are about 20 positively charged residues in KCSA, and we mutated those residues one by one, and look at this channel activity. You see this mutant shows lower open probability, and they all have located in the N-terminal part of the channel, such as R11, K14, or this is the deletion of the 22 residues from the N-terminals. By deleting or by neutralizing these positively charged residues, the open probability is decreased. So the N-terminal part, which is now called the MO helix, should interact with the head group of the negative charged phospholipid in the inner leaflet of the membrane. This is a result of the EPR predicted structure, and this is a N-terminal MO helix. But the crystal structure, even there is the N-terminal part, it is not resolved. And even it is called full-length structure, the N-terminal MO helix has not been resolved. So people do not interested in the MO helix, but we found from the mutational experiment, the MO helix should be very important. This is a fluorescence labeled experiment, and to see the importance of the residue. Making the long story short, we just show the result. From the residue 10 to 17, the tetramethylrhodamine is labeled one by one, and this signal shows the difference of the fluorescence intensity in the activated channel and the inactivated channel. And this is a pattern, periodic pattern. It is 3.6 per turn. So it means that this part forms the alpha helix. And as the helical field shows, this is typical amphipathic helix. And in this part, there are two positively charged residues. So from this result, we just skip the discussion in detail, but we proposed roll and stabilized model. This MO helix stabilized with the negatively charged head groups with positively charged R11 and K14 in an open conformation, but it revolves around the axis of the helix in a closed conformation. So the MO helix may rotate around the helix in the open closed conformation. This is our predicted changes in the MO helix. Now we show the other benefit of the bubble CBB contact bubble bilayer method. We can change the solution rapidly by introducing the micropipette and inject aqueous solution. We can change the solution within 20 milliseconds. The KCSA channel is a pH sensitive. So when the intracellular side is changed from neutral to acidic, from acidic to neutral, the channel activity immediately abolished and returned when the pH returned to neutral. So we can perform this kind of perfusion experiment similar to the patch clamp experiment. And considering this organization of the contact bubble bilayer, we found several rules of this system. This is a bulk organic solvent, and this is a monolayer, and this is a bilayer. Rule number one, each leaflet of the membrane is an open system. It means connecting the one leaflet of the bilayer. The lipid in this monolayer is diffusible into the one of the leaflet of bilayer. Rule number two, the bulk organic solvent is contiguous to the membrane interior. This is very interesting architecture of this method. I will show you later. And also the membrane lipid or integral membrane protein can diffuse on the bilayer, but it is restricted in the bilayer, never move to the monolayer. These three rules is very important to do the next experiment. We can inject the hydrophobic substances around the bilayer, but in the bulk organic phase. Then the organic substance may diffuse or partitioned into the monolayer or bilayer. So during the channel recording, we can change the lipid composition. This is another type of the perfusion. We call it membrane perfusion, which has never been performed before. And this is, the paper is accepted yesterday. You will see the detail. This is the example of KCSA. You see several channels is keeping active in the steady state. But when the cholesterol is perfused, then the channel activity immediately abolished, and it is returned. So we can see the immediate response of the hydrophobic substance. It is just simply the cholesterol, but we can use many other types of the hydrophobic substance in this method. Just already time. I need to show one experiment. This is a KCS channel resolved in the FM. You see it is only 3 nanometer, but you see tetrameric structure, 4 subunits, and there is some dip in the center. This is the usual our AFM measurement, but it is important to fix the orientation of the channel to see the outer surface or inner surface. To do that, we developed a method. Usually, channel incorporated liposome is spread on the mica, but we changed the sequence of the experiment. First, we modified the mica surface with nickel, and the channel is attached beforehand. For the solvulus channel is attached through the histag. Then liposome is added. But it is important to use a destabilized liposome. It means the liposome contains detergent. Then you see the filling of the membrane. You see these particles represent the ion channel, but as the liposome perfuses, the interspace is filled, and eventually all the space is filled by membrane, and the channel is incorporated in the membrane with the same orientation. It is the KCS channel has a site for the azitoxin channel. So by using the azitoxin, you see the azitoxin has already solved the structure, and this is unbound channel. You see the tetrameric structure, but once the azitoxin is bound, there is a bulk substance. So by measuring the height of the channel, you see the bind and unbind kinetics from this kind of experiment. We don't have time to show the gating coupled clustering dispersion dynamics, but those things are also performed using the high-speed AFM. This is another type of the single channel interacting each other on the membrane. This is my group. Thank you for your attention.
We have questions. So I have a rather odd question, but what's known about the difference between lipid composition in Streptomyces viridans between the two sides of the membrane, the inner and outer leaflet? Do you know much about that?
On the bacteria?
Yeah.
Not so many information exists.
Yeah.
But PG is rich in bacteria, but no information on the asymmetry.
Yeah.
But the usual eukaryotic cell, PS exists in the inner leaflet, but PC exists in the outer leaflet. Yeah. So that is important.
Very nice work. I wasn't aware of it. So you were focused on the head groups, but is there any regulation by the acyl chains also? Because it looks like you would be able to address those questions also.
Yeah, that is a very good question. We have performed the experiment with a different type of the tail. Branched tail group, such as phytanoil group, which is popular in the archaea. So by using this branched tail, comparing to the usual tail, we see the different activity of the KCSA channel.